Thank you very much, Lisa. Yes, as mentioned, my name is Kalina Grabb, and I am a 2023 Knauss Fellow in the Ocean Acidification Program Office at NOAA, and I deal with international policy. So it is my honor today to introduce Colette Fletcher-Hoppe, and Colette is a PhD candidate in biology at the University of Southern California in Los Angeles, and her research focuses on the structure and function of marine microbial communities, such as bacteria, viruses, and single-celled plants and animals that make up the bottom of the marine food web. Colette is particularly interested in studying the ways in which climate change alter these communities over space and time. And so with that, I'd like to welcome Colette to give her presentation, which is entitled Spatial and Temporal Dynamics of Marine Microbial Communities in the Southern California Bight. Take it away, Colette.
Thank you so much, Kalina. And just to clarify, are you able to see and hear me okay?
Yes, you look great.
Great, thank you. All right, thanks so much, everybody, for joining me during your lunch hour. As Kalina mentioned, my name is Colette Fletcher-Hoppe. I'm a PhD candidate at the University of Southern California in the Marine and Environmental Biology Department, and my dissertation focuses on the spatial and temporal dynamics of marine microbial communities in the Southern California Bight. So to get things started, I thought I'd begin with a little bit of an outline, breaking my title down into its component parts. So on the left-hand side here, we have my title, and on the right-hand side, we have an outline of what I'll be talking about. So I'm going to start with some background on marine microbial ecology, then move into fine-scale spatial dynamics, then into longer-term temporal dynamics of one particular organism, and wrap up by talking about some future work that I'll be doing over the summer. So to start, let's begin with some background information, including what marine microbial communities are. So these are networks of single-celled viruses, bacteria and archaea, and single-celled algae and protists. And some of them look like this. So to clarify, this is an artistic representation of a community. It's what's known as a diatom mandala. So somebody has taken, excuse me, microscopy images of single-celled plants and animals.
Colette, you need to, just go ahead and forward that because we're still seeing the outline.
Is that showing up now?
Not yet.
Uh-oh.
There it goes. There it goes. Perfect.
Okay, thank you for letting me know. Okay, so these are networks of single-celled viruses, bacteria, archaea, and protists that live in the oceans. And so the image here on the left is a composite image of many different microscopy images of some of the single-celled plants and animals. And as you can see, they're beautiful, right? This looks like looking into a kaleidoscope. And there are thousands of these living gems in every milliliter of seawater. And importantly, just like the plants and animals on land, these organisms have complex interactions with one another. So some of them do photosynthesis. They take in carbon dioxide, they produce oxygen. Some of them are predators like dinoflagellates. There can be symbioses, there can be parasites like Syndynialis, and they all compete with one another for resources. And of particular importance to the NOAA folks in the room, they are the base of the food web of the marine ecosystem, so they can be fish food. Another really important function of these marine microbial communities is that they serve as an important carbon sink to mitigate the effects of climate change. So as I mentioned, the microbial communities can do photosynthesis just like plants on land. They take in carbon dioxide and produce oxygen, as shown in these two figures here. So the figure on the left is showing chlorophyll concentration in the different regions of the ocean, and that's a proxy for microbial biomass, or how much carbon is in the water column. So you can see there's really high concentrations of carbon dioxide at the poles and along the equator, which means that these organisms here are taking in carbon dioxide at high rates in these regions. And in turn, they're producing oxygen in the water. So the map here on the right shows the concentration of oxygen in the oceans. And as you can see, it's again really high in the poles and along the equator. So in other words, these organisms are taking in carbon dioxide, they're producing oxygen, just like plants do on land. And similar to plants on land, this process relies on fertilizer. so sources of nitrogen and phosphorus. There are many ways to study these marine microbial communities, but my lab chooses to focus on DNA sequencing. So that process kind of looks like the outline that I've illustrated here. In the first step of our methods, we collect some samples. So we'll go out to a particular location on a boat, bring up a lot of seawater, and then we'll filter it. So this works similar to your coffee filter, for example, or a filter to purify your water, right? You have seawater going through a specialized piece of paper, and then organisms of a particular size class get stuck on that paper. We can choose what size organisms we want to stick to the paper. And then we'll do DNA extraction, so taking the DNA out of the whole community that's stuck on our filter. And we'll sequence the DNA and use computer code to do models, statistical analysis, and to process the DNA sequences into something resembling a species. Sometimes computer coding is a little frustrating, a little challenging, and you just got to take a minute to bang your head on the desk. But hopefully that part only lasts a minute, and then you can get into the fun stuff. So my lab chooses to focus these methods on the Southern California Bight. And for those of you that don't know where that is, it's this region right here, indicated by the red circle. So this is the part of the California coastline where it looks like somebody has taken a bite out of the coastline, although bite and bite are spelled differently. And there are many reasons why we want to study the communities here, but one of them is that the Southern California Bight has these really interesting complex current dynamics. So one of them is seasonal upwelling. So every spring on an annual basis, wind patterns will bring nutrient-rich cold seawater to the surface, and that fuels a big boom in the microbial community's population. So I mentioned earlier, microbial communities are able to do photosynthesis and all of these processes that we see on land, but they need fertilizer in order to do so. So essentially you can think of upwelling as bringing in fertilizer into the region, that cold, nutrient-dense water. And that prompts the microbial communities to start growing and competing with one another and being really active on an annual basis, which in turn fuels a thriving fisheries economy off the coast of Southern California. So for this reason and for many others, my lab focuses our sampling efforts here. And I'll talk a little bit more about this green dot in the next slide. The green dot represents the location of our time series, the San Pedro Ocean time series, which I'll be referring to as SPOT conveniently throughout the rest of these slides. So my lab and some other labs have been going out to this location once a month for the last 20 years. And every month we take a suite of abiotic measurements, including nutrient availability, temperature, salinity, oxygen, and we also do some cell counts to get an idea of the size of the microbial community. And we do our sequencing technique to look at the relative abundance of different bacteria, eukaryotes, and viruses. And the filters that we use, the schematic looks a little bit like this. So we have an 80 micron nylon mesh to remove larger organisms such as fish or kelp or whales or whatever we don't want to see in our marine microbial communities. And then our larger filter is going to be 1 micron in pore size diameter. So every organism between 1 to 80 microns in diameter is going to get stuck on this filter. And then the smaller size class of organisms is captured by a 0.22 micron filter. So our smaller size class of organisms is everything between 0.22 to 1 micron. And this is a really invaluable resource because it allows us to see how the entire community of these two size fractions has varied at this location for every month for the last 20 years. So now that I've covered some of the important background information, I'll start talking about my first chapter, which is looking at spatial dynamics of the marine microbial community. And one of the big takeaways of our San Pedro Ocean time series dataset is that over time, the larger size fraction of organisms is less similar or less stable within itself as compared to the smaller. So here I'm using a metric of similarity called Bray-Curtis similarity, and that's going to be a sliding scale from 0 to 1. So if 2 pairs of samples score a 0 on this scale, that means that these 2 samples could not be more different to one another. And if the 2 samples score 1, that means that they're identical. So a score of 1 means that the 2 samples have all the same organisms in all of the same proportions. A score of 0 means that the 2 samples have no organisms in common whatsoever. And here we have Bray-Curtis similarity plotted on the y-axis and time on the x-axis. And you can see this really interesting oscillating pattern where every 6 months the community is alternately really similar or really dissimilar, or really similar or really dissimilar. So it goes through this regular cycle every year. And the thing that I want to draw your attention to most is that the smaller size fraction here is consistently more similar to itself than the larger size fraction. So it scores a Bray-Curtis similarity score of about 0.55, and the larger size fraction scores about 0.4. So that's how the community is changing on a monthly basis over time. But I wanted to see how representative these changes are of the larger ocean. So in order to do that, I started looking at spatial diversity. So to explain that a little bit more, I'm asking the question, how does the whole community change at multiple locations? And that in turn will tell us how representative the community is of or our time series data is of the larger surrounding area. So if all of the stations in my transect are identical to each other, then we can say spot represents this whole transect accurately. And if they're very different to each other, then we know that spot is kind of a unique location. So in order to look at that question, I followed the following sampling methods with some help from collaborators. So collectively, we sampled 9 different stations in kind of an X-shaped pattern around spot. So in this case, yes, X does mark the spot. And so once we took our samples, we followed our standard filtering schematic where we have captured communities between 1 to 80 microns here, and our smaller community is going to be 0.22 to 1 micron here. We did DNA sequencing and DNA extraction of all of these communities, and then we calculated Bray-Curtis similarity and started looking into some statistics, such as the following. So here we have a graph of Bray-Curtis similarity on the y-axis here, but in contrast to the graphs that I showed you previously, we have space on the x-axis instead of time. So the different points on this graph represent the distance between the sampling sites. So all of these box plots here at 0 kilometers represent replicates. So samples that were collected from the same site at the same time. Samples that are in the 3.2 category box plots, those represent pairs of samples that are 3.6 kilometers apart from one another, and so on and so forth. And we can see that Bray-Curtis similarity in the smaller size fractions plateaus at about 0.65 within 3.6 kilometers of sample collection, whereas samples in the larger size fraction plateau at about 0.55 within 4 kilometers of sample collection. So essentially, the smaller size fraction is more similar to itself over space, and the larger size fraction is a little bit more distant to itself over space. When we plot that in a different way, such as on a map, we see the same trends again. So these are the same Bray-Curtis similarity data that I just showed you, but they're plotted on a map instead of in a bar plot spaced out. So here we're comparing Bray-Curtis similarity to our time series location, which is again in the middle of this X-shaped transect. So a score of 1, or these cooler colors here, indicate that all of the samples collected from spot are very similar to each other. And as we move away from spot, the samples become more distant as indicated by the warmer colors and the higher Bray-Curtis similarity scores. So, or the lower Bray-Curtis similarity scores, excuse me. So the large size fraction here is on the left, and as you can see, as we move away from spot, the colors become a little bit warm, but they become much warmer, much more quickly in the larger size fraction. So in other words, as we move away from spot, the larger size fraction of marine microbial communities becomes less similar more quickly. And I wanted to know why that was for the larger size fraction. In other words, I wanted to know what types of microorganisms are driving this drastic difference. So I pulled out the microbial species based on their metabolic function. So this panel here, for example, is all of the archaea in the larger size fraction, so the 1 to 80 micron group only. And we can see that the samples, just based on archaea relative abundance, are pretty similar to one another, represented again by those cooler colors. But the photosynthetic organisms, in contrast to the cyanobacteria, are very different or very patchy. So we have some sites with only looking at cyanobacteria that look very similar to spot in the center, but some sites indicated by these red colors here are quite dissimilar or very distinct. So to wrap up the conclusions of Chapter 1, we can see that Bray-Curtis similarity plateaus at about 5 kilometers for both the smaller and the larger size fractions. But both the smaller size fraction and the larger size fraction are above 0.5. So that indicates that the community is pretty similar to itself over space, but there is more of a patchy distribution of the larger organisms and a homogeneous background of smaller microbial organisms. So to move on to my next chapter, the first chapter was looking at spatial variation in the whole community. Next, I wanted to look at temporal variation of one specific organism that I think is really cool and interesting. So here I'm looking at temporal diversity of the organism UCYN-A. This is a really interesting organism for many reasons. The first is that it lives in obligate symbiosis with a eukaryote called Brachysphaera bigelowii. So essentially, these two organisms are found together quite often because of UCYN-A's unique metabolism. UCYN-A is, for those of you photosynthesis nerds, it's missing photosystem 2 and TCA. I think a good metaphor, if you envision UCYN-A as a person or a larger organism like an animal, it would be missing one lung and its entire stomach, which is crazy. Like, how does an organism live that way? Well, the answer is that it has this really close relationship with Brachysphaera bigelowii, as depicted in this schematic here. They are constantly interchanging metabolic compounds with one another. They're physically associated with each other, and as you can see, they're interchanging all of these different compounds. So it kind of doesn't matter that UCYN-A has a stomach because its buddy Brachysphaera bigelowii is there to feed it. And this relationship has broader global significance too. So as you can see in this schematic, UCYN-A is exchanging nitrogen compounds for carbon compounds. And to go back to some of the information that we discussed in the background section, again, here we have the chlorophyll concentration of all of the marine microbial communities in the ocean. And I told you that is contingent on nitrogen compounds, as indicated by this map here. So we have a map of the global nitrogen concentrations throughout the surface of the ocean, and it closely mirrors that of chlorophyll concentrations. So where you see higher biomass, you see higher nitrogen concentrations, or the other way around. Where you see higher nitrogen concentrations, you see higher microbial biomass. So essentially, one of the reasons why UCYN-A is so specific or so important globally is that it's able to provide its host and other organisms that interact with the host access to these nitrogen compounds. Essentially, it serves like fertilizer. UCYN-A is a group of several rare organisms that are able to produce nutrients out of thin air, essentially by taking nitrogen gas and making it into a bioavailable form. And so these organisms can fertilize the whole community on a larger scale. UCYN-A has multiple different types, each of which have a broad global distribution. So UCYN-A1, indicated by the blue colors on the top panel of this map here, is going to be our open ocean ecotype. It's a little bit smaller and it has just 1 or 2 symbionts per host. UCYN-A2 is represented by the red dots on the panel B of this map. And you can see it's our coastal ecotype. It's really prevalent in coastal waters. It's larger and it has a larger genome and multiple symbionts per host. So UCYN-A types are found throughout the ocean, and you'd sin if you thought they weren't found in California. They are. So to answer some questions I had about UCYN-A and its autocology, I turned to our San Pedro Ocean Time series. So first I wanted to ask, how specific is the partnership between symbiont and host? And I looked at the relative abundance of UCYN-A type 1 to answer this question. So indicated by the gray dots here, UCYN-A1 relative abundance is plotted over time, and the green triangles here indicate the relative abundance of the host organism over time. And you can see that anytime UCYN-A1 is present, its host is present also. So they're always found together in a pretty much constant ratio. But things look a little different when you look at the relative abundance of UCYN-A2, which is given by these gray triangles here. So when we look at the relative abundance of UCYN-A2 over time, we can see that there are several sampling dates where UCYN-A2 is present and its known host, indicated by the blue squares here, is absent. And I was particularly interested in this state here, where UCYN-A2 is present and the known host is not present, because of another variant closely related to the host, indicated by the purple circles in the lower graph. So you can see on this state, the symbiont is present, the known host is absent, and a close relative of the host organism is present in high abundances. So this to me suggests that UCYN-A2 could have multiple hosts. There are days where the symbiont is present and a different host is filling in that metabolic relationship, in contrast to the one that is more commonly studied. So the next question that I wanted to answer is, with what non-host taxa does UCYN-A interact? So in order to study that, I looked at an extended local similarity analysis, which basically asked the question, what taxa co-occur on the same day at spot? And as you can see, based on the results that I plotted up here, a lot of dinoflagellates are co-occurring with UCYN-A. And remember, I mentioned that dinoflagellates are predatory organisms. And I was particularly interested to see this dinoflagellate circled in pink. This species is a member of the genus Lepidodinium, which has been hypothesized to be a predator of UCYN-A. So the fact that we're seeing it co-occur with UCYN-A at spot indicates that it might actually be eating UCYN-A and its host organisms. I was also really interested to see Symbiotic here, which again, as I mentioned, are parasites, but they're known to parasitize dinoflagellates. So based on the organisms that are co-occurring at spot, I'm hypothesizing the following food web, where we have Lepidodinium and other dinoflagellate ASVs eating UCYN-A and its host, and Symbiotic parasitizing those organisms. So next I wanted to look into what factors govern the presence of UCYN-A at spot. And so in order to do that, I looked at the average of certain abiotic parameters on dates that UCYN-A is present or absent. So in the top row here, we have UCYN-A1, and in the bottom row here, we have UCYN-A2. This is our open ocean ecotype. This is our coastal ecotype. And I was really interested to look into those unique characteristics of the Southern California currents. And in particular, I wanted to look into upwelling. So again, this is a seasonal occurrence where nutrient-rich cold water is brought to the surface and spurs a big increase in chlorophyll concentrations. So you can see on dates that UCYN-A1 is present, upwelling is much lower. And on dates that UCYN-A1 is absent, upwelling is much stronger. And similarly, temperatures are higher on dates that UCYN-A1 is present, indicating no upwelling is occurrent, and they're lower on dates that UCYN-A1 is absent. And it's, again, chlorophyll is higher, or chlorophyll concentrations are lower on the dates that UCYN-A1 is present, indicating that upwelling is not happening, and higher on dates that UCYN-A1 is absent. And the same trends kind of holds for UCYN-A2, but they're not statistically significant. So remember, again, this is our coastal ecotype, and we hypothesized that it's not strongly influenced by upwelling dynamics because it lives in the coast. It is not deterred by upwelling because it's just accustomed to those kind of conditions. And this question becomes important when you consider the fact that upwelling is predicted to intensify with climate change. So that begs the question, what will happen to UCYN-A presence at spot? Will it be deterred from being present at this location? Results are mixed, to say the least. So to conclude this portion of the talk, the specificity of the UCYN-A partnership depends on the type, where we see type 1 co-occurring with its host pretty faithfully, and it looks like type 2 is able to co-occur with multiple different hosts. UCYN-A and its host might interact with dinoflagellates, including Lepidodinium, a potential predator, and Symbiotic as well. And UCYN-A1 distribution is strongly influenced by upwelling indices. So moving on to some future work, and I'll conclude with acknowledgements to hopefully allow some time for questions. My future work is going to try and look at what UCYN-A is doing off the Southern California Bight. So this is some preliminary data. For the sake of my collaborators, I'm not going to show all of it, but we do see, based on the transcriptome of UCYN-A, it's expressing genes that are used to make nitrogen bioavailable. So that kind of begs the question, are other organisms doing this? And will it be important? Because as I mentioned, upwelling continuously brings these nitrogen resources into the Southern California Bight. How much is additional inputs of nitrogen influencing the rest of the microbial community? And as climate change continues to influence ocean currents, will this become a problem? That's going to be some of the work that I'm looking into in the future. So I want to conclude with some acknowledgements. I want to say thank you to my thesis committee members, including Jed Furman, my chair. Thank you so much to the rest of my lab mates for their support. And thanks very much to the Knauss and my host office, the Department of Energy, for supporting my work there and allowing me to learn about some new topics. And lastly, thank you so much to Kalina and to Lisa, the seminar organizers, for giving me the opportunity to speak. I really appreciate it. And I think we have some time for questions.
We do. Thank you so much, Colette. This was wonderful. Audience, we have about 4 minutes for questions, and so please type them in the questions chat box, and I will read them to our speaker. But while we're waiting for your questions, I encourage you to download Colette's slides, which are attached to the control panel under handouts. So you see her initials, CFH, and you'll be able to review her slides on your own time. And one last reminder as we wait is if you know of someone who might be interested in Colette's research that was not able to attend live today, I encourage you to let people know about the recording, which we will be housing on our NOAA Central Library YouTube channel in just a few hours. I will go ahead and put that link in the chat box. I put it in earlier, but just in case you joined us late, you'll see it in there now. So Colette, thanks. We're going to wait another minute or so. Here's a question. It says, thank you, Colette. I'm curious how you expect these dynamics with UCYN-A and other organisms in the California Bight to be similar or different to other regions of the oceans.
That's a great question. And to answer it, I think I'd like to pull up one of my previous slides showing the concentrations of microbes and nitrogen concentrations. So let's use this for an example. Okay. So climate change essentially is expected to intensify the dynamics that are already present in the ocean. So California, for example, experiences seasonal upwelling that brings nutrient-rich waters to the surface, and that's expected to intensify with climate change. But so too are ocean currents that govern these areas where there's low microbial biomass and low nitrogen concentrations. So organisms such as UCYN-A that are able to bring more nitrogen into the system might become more important in these nitrogen depleted regions with climate change. Whereas, as I showed in one of my later slides, here, climate change may exacerbate upwelling and deter UCYN-A from being present in this region. However, that gets a little bit more complicated when you consider El Niño, which is an interannual event that occurs in the Pacific Ocean that has an influence on upwelling dynamics as well. So when I say the answer is unclear, that's because there are a lot of complicated factors at play. If you'd like to talk about this more offline, I would certainly be happy to do so. I think it's a really interesting question.
Wonderful, thank you. We have one other question. This person asked, awesome presentation, Colette. What is your hypothesis as to what is driving the differences in BC dissimilarity between large and small size fractions? Are there overall differences in gamma diversity between the sizes?
That's a great question too. So when I was presenting that Bray-Curtis dissimilarity data comparing the smaller and the larger size fraction, we saw that there's a homogeneous distribution of the smaller organisms in comparison to a patchy distribution of the larger organisms. And I think there could be many reasons for this. So just to clarify, the fact that the smaller size fraction community is more similar to each other does not mean that there are fewer species. There are actually more species in the smaller size fraction, but they're more homogeneously distributed. And I think that kind of goes back to, I'm gonna just jump here. Ocean currents as represented by the black arrow on the map here. So this black arrow, in case I didn't specify earlier, represents the prevailing direction of the currents on the dates that we took our samples. And my hypothesis is that the smaller size fraction is going to be homogeneous over our sampling site, but ocean currents are bringing in particles with the larger organisms attached to them, which might be more influenced by the flow of water.